Now we'll move on to the application portion. And so I'd like to introduce Dr. Alessandro Sette back, who you may have heard from day one if you had joined then, our co-PI of the IEDB. And Alex will be presenting the CEDAR prostate cancer meta-analysis. So thank you, Alex.
Thank you, Nina, and get well, Zeynep. So what I'm going to do is present you the outcomes of the meta-analysis that we did on prostate-specific antigens. And this is quite different and by design from a lot of what Zeynep has been talking about in terms of neoepitope and mutated epitopes. The first bullet doesn't really need to go, we don't need to go through. We've already been talking and introducing the cancer epitope database and analysis resource, the sister database to the IEDB. And as alluded to in the second bullet, while we obviously are still developing the cancer-specific aspects of CEDAR, we also started to curate and populate the CEDAR database. And we decided initially to focus on two prototype categories. One is the neoepitopes. So these are mutated epitopes. And as mentioned earlier, typically these are very cancer-specific in the sense of a particular patient and particular tumor instance specific. And prostate cancer antigens, which is instead the orthogonal distinct data source in the sense that the prostate antigens are typically cold tumors. They don't have a lot of mutations. And these are well-defined cancer antigens, which resemble regular antigens. So it's a protein. And so we wanted to look at this data as in parallel with the meta-analysis is developed instead for the neoepitope mutated antigens. And the goals are, as usual, in the context of a meta-analysis, it's interesting, and we can use this to illustrate the capabilities of CEDAR and solicit feedback, and also realize what things maybe need to be optimized or developed in the context of CEDAR, but also specifically look at the domain of what is available in the scientific literature related to epitopes derived from prostate antigens, and highlight strengths and highlight knowledge gaps. So what is a prostate-specific antigen in the first place we need to answer? As we have been highlighting in earlier today and in the previous day, we have a process where we identify potential cancer-related papers, and then we can filter down, and about 11,000, a little more than 11,000 at the end of 2022 were looked at, and about 400 were related to prostate-associated antigens. Essentially, we curated most of them. And after reviewing the abstract, 96.5%, and after reviewing the abstract, about 165 were found to contain curatable information. And this is not different from what you see in other instances, because our process to capture information, it's by design very broad. So we don't want to have false negatives. It's okay to have a false positive in the sense of a paper that originally was thought it might be curatable, but then is under closer inspection is found not to contain curatable information. So we want to be comprehensive in our strategy. And then we focused on what are prostate-specific antigens. We really did our homework, we think, looking at the scientific literature and identifying 8 prostate-specific antigens. And so this is an important point in this meta-analysis, in the sense that a prostate cancer may express some of the prostate-specific antigens, such as PAP or PSA, and we'll get to that in a second, but may also express other antigens, such, for example, MAGE, or other antigens, which is really not specific of prostate cancer. These are cancer antigens that may be found in prostate cancer, but also breast cancer or lung cancer, and so forth. And so here today, what we're really focusing on are these 8 antigens, which are exquisitely specific for prostate cancer. And we're excluding these other antigens, which are more broadly found in prostate cancer, but other cancer types as well. So by now you're familiar with the CEDAR homepage, with the main 6 search panes, which are very similar to what we have in the IEDB. And so when we do the search for the prostate antigens, here you have this is an example with PSA. It's the same structure with the tabs showing the epitopes, the antigens, the assays, the receptors, and the references. And as in the IEDB, you can adjust the query parameters by changing what is entered in the various panes. And so here if you click on the assays, again, similar to what you see in the IEDB, you have these different columns that detail what was the particular assay for that particular epitope, that particular reference, that particular instance. So what did we find? What did we do in terms of the prostate-specific antigens? So here is the list of the prostate antigens, and so there is the PAP, or prostatic acid phosphatase, PSA, the prostate-specific antigen, PSMA, PROS, the prostate stem cell antigen, PSCA, STAP, TRP8, TARP. You see here, there's about 132 papers related to these 8 antigens, and already see that 3 antigens here really have a lion's share of the references related to this set of antigens. In terms of a grand total of epitopes, is about 300 epitopes in total, with about 278 related to either B or T cells, and about 93 being related to binding, MHC binding or MHC elution. You see, if you look, just looking at the grand totals here, it's about a 2-to-1 ratio of epitopes defined in a human versus the mouse host, and a fairly balanced representation between B and T cell epitopes. When you look at the T cell epitope, again, it's about a 2-to-1 ratio of class 1 to class 2. Far more MHC binding than MHC elution, 93 to 21 total epitopes defined. And as I was saying, these 3 antigens have, these 4 antigens have the lion's share of the total epitopes, and is you can see perhaps, as also we'll discuss later, depending on the specific antigen, you might have more of one type of epitopes or so forth. And this is the first bullet, re-summarize what I just said. We looked at the number of epitope as a function of the protein, as a function of the number of publications and protein length. And this was based on also analysis we've done in other systems. And so basically what you find is that there is a very significant correlation of the number of described epitopes correlating with the number of publications, but no real correlation between protein length and epitopes. And well, at saturation, you expect that in general, bigger proteins have more epitopes. But the fact that you don't really see that, but you see a correlation number of publication, it means that we are far from having saturated. The literature has not really captured all the epitopes that are out there, because every new publication comes up with a bunch of new epitopes. So there is a lot to do in terms of the scientific community defining prostate antigen-derived epitopes. So what about T-cell assays? If you look at the T-cell assays, you essentially have enough assays to really do a meaningful analysis of only for the four main antigens, PSA, PSMA, PAP, and PSCA. And here you see that most of the antigens are T-cell assays, but when you look at the number of assays that actually use tetramers, which is the gold standard of what you use for assessing functionality of T cells, there is really a few for PSA, but very, very little else for other antigens, and particularly in the case of class 2, there is really almost nothing in terms of available assays that have been taking advantage of tetramers. And yeah, that's an important point. And the other point is when you look at the MHC ligand, either elution or binding, again, there is a very large bias in terms of what is recognized in the context of class 1 versus class 2, with very much less so available in terms of the class 2 as compared to class 1. Further, when you look at the restriction, there is a tremendous bias, particularly in the case of class 1, whether you look at T cell or MHC binding assays, it's a vast majority of what's out there is for A201 or A2402. And other alleles are very, very poorly represented. It's a little bit better when you look at class 2, but still the number of T cell assays that have a defined restriction are very much overrepresented in HLA-DRB 0101, really implying that there is a lot to do still in defining epitopes for other alleles, particularly because, as we know, certain alleles are very are differentially expressed in different ethnicities. And so this is really a snapshot that is mostly A2 is mostly frequent in Caucasians. This is an allele that is frequent in Asian descent populations, but as I said, there's very little else. What about the type of assays in cytokines? And this is really striking. If you look at the cytokines that have been measured, it's basically very much all interferon gamma. And the other cytokines have not been studied very well. And really, this is another knowledge gap, because it is known that polyfunctional T-cell responses are important in providing protection and in providing anti-cancer activity. In terms of the B-cell epitopes, if you look at the kind of high level, there is a fair representation of both monoclonal and polyclonal. In PSA, there is more monoclonal data. In PSMA, it's the other way around, and the same for PAP and PSA. But overall, it's a fair, similar representation. When you look and you drill down at the type of assay, again, mostly ELISA and very little of other assays, such as crystallography or biological activity or immunostaining and so forth. Another knowledge gap that we noted is that despite the fact that in the cancer setting, really to show that the T cell that is associated with a particular epitope is capable of recognizing and killing, recognizing or killing a tumor cell, there's very little, again, in terms of antigen assays that have been described involving recognition or killing of tumor cells, and the numbers are shown here. Likewise, there are very few assays involving in vivo epitope assays. And so here you see really a scatter, and then this is perhaps a reflection of the fact that there are differences in anatomy between humans and mice for prostate, and so in vivo assays are not as easily performed. This points out to the fact that it's very, very little available in terms of TCR1, BCR3 sequences. So again, lots to do for the scientific literature. And this is particularly, as summarized here in the last bullet, the fact that you have 1 TCR and 3 BCR is, if you compare with what is available for COVID, where you have 87,000 TCRs, it really brings home the point that there is a lot to do. One other point in terms of the CEDAR, we classify the type of disease associated with the cancer. And this shows that despite the fact that there are many different grades and types of prostate cancer, that information is not very adequately represented. It's not because we don't want to curate it, but the papers really don't often provide that level of granularity. Finally, Nina, do I have 2 more? Yes, okay, we're good. Okay, so finally, a point that I made earlier on, that these prostate antigens really are regular antigens with well-defined protein and sequence, and with a lot of epitopes derived from that particular antigen. That allows us to visualize the T cell and B cell activity with immunobrowser along with sequence. And so this is an example of for PSA. And what we showed here was a class 1, class 2, and B cell epitope distribution and frequency. And why is this? I mean, this, first of all, is interesting to see that there are domains and regions of the proteins that are more dominantly recognized. But also, if you look, for example, for an application where you might want to use only one subdomain to simplify process of an immunotherapy application or whatever, this would reveal that, for example, using this region would be good for both class 1 and class 2, but this region here would be great for class 2, not so much for class 1. And likewise, if you're looking for something that would be able to induce both helper T cells and antibody response, you would probably focus perhaps on this particular region that also would be devoid or not particularly rich in class 1 activity. So there are comparative analysis that can be done and guide further developments and analysis. And so the overall conclusions, briefly summarize that we have concluded, essentially, we're now in steady state, the curation of prostate antigen papers. This is now a wealth of searchable information for the scientific community. We also identified several different knowledge gaps, amongst them the limited number of prostate epitopes for which there is TCR, BCR receptor data available, and very limited number of in vivo and tumor recognition assays. Also, we suggested areas for further improvement and expansion of CEDAR. And finally, the analysis of immunodominant regions has potential implication for vaccination or immunotherapy strategies. And this is now published. And so if you want, you can read all the gory details in there. And with that, I can stop sharing my screen and be back in here.
Awesome. Thank you so much, Alex. That was great. I'll give you a couple of minutes to have a look at the Q&A because we do have some questions in there for yourself and Zeynep to tackle together. Now, Zeynep, I know you pushed through the 30 minutes, but I would like to invite you back for the Q&A with Alex. Would you like to take the first question?
Okay, I can pretend to be Bjorn and answer the question from Dinong Wang that says, thanks, Bjorn. For the cancer epitope, is there a plan to cover tumor-associated carbohydrate antigens and epitopes? Yes, certainly we are well aware of the wealth of information related to carbohydrate antigens and, TN and also the gangliosides and all that. There's a whole literature there. So we are following them. Right now, we are focusing on more of a on the peptidic antigens, and depending on also available resources in terms of curation capabilities, we certainly will tackle the non-peptidic carbohydrate antigens as soon as we can.
Okay. Hur Al-Hassani asks, maybe I missed it, but when are the where are the mutations taken from? So the mutations, we take the mutated peptides directly from the peptide that has been published. So the researchers, they do like this cancer analysis. They do, for example, sequencing sometime to detect those mutations, and then they test peptides, and that's how they end up with us. So they test some of those mutated peptides, and then we curate them directly how the researchers have published them. And we can then go back and see where it was taken from this neoepitope, from which antigen, and then we can compare it to the wild type peptide and see what the specific mutation was. Yeah.
So I'll jump on a question from Flavio. Flavio says, however, a promiscuous neoantigen is very interesting to general cancer screening. Do you agree? Yes, we totally agree. And actually, this is a very important aspect for the field in general, in the sense that clearly, the patient-specific neoepitopes are of interest, as well as the non-mutated cancer epitopes, such as the prostate antigens. But there is this intermediate area, and one of the things that the meta-analysis that Zeynep is performing is going to actually highlight that there may be certain antigens which are frequently mutated, and so that they could be, and for example, KRAS is an example of that. So there are antigens that are very interesting, and also there may be there are mutations that are found recurring in different patients, and that is also obviously of real interest from an application point of view, and that is one of the powerful outcomes of CEDAR, to be able to pinpoint these instances.
I'll answer Jason Pereira. Will the DNA-based information for epitopes also be available via the database CVS download for CEDAR? Yes, absolutely. So whatever we add to CEDAR, the website, it will also be available in the export. So when you download the data, you will also be able to access those data sets. In terms of the Open Cravat link, maybe we'll just put the link in there. But if you want to have all the details that Open Cravat has found while annotating the mutation, you would then have to follow the link and get the data from the Open Cravat page. But the most important information that we deemed important will be directly added to CEDAR and also available in the downloads. I can also answer the next one. Are you trying to annotate those non-synonymous SNPs? I'm not sure what is meant here, but I think, yeah, so non-synonymous SNPs, they generated the neoantigens, right? So if the DNA mutation is non-synonymous and it's in a coding region, it will generate a mutated peptide. So we start from the peptide basically, and we want to translate it back to the DNA mutation. And yeah, that's exactly what we are doing. I don't know if there's a follow-up question. Feel free to ask. I think that we're all.
I think we're done.
Definitely looks like we're done. So that's awesome. And we're running about 10 minutes ahead. So that just means we get a slightly longer break, which will probably be nice for everyone. So a massive thank you again to Zeynep and Alex for the wonderful presentations and how it can be used in practice. So thank you both.